All right, good morning, everyone. As Jason said, I'll be starting at Stanford in the Department of Genetics on July 1st. So to give you a high-level overview of what I'm interested in, I'm very interested in controlling cell behaviors. So I'm very interested in controlling cell behaviors, and those behaviors include things like survival, proliferation, differentiation, and in the case of immune cells, cytotoxicity. The way that I like to think about controlling them is through proteins called receptors, particularly modular receptors, like CARs and CCRs, but also cytokine receptors and synthetic receptors like ChEM-H. And the way that I like to study these is through library screening, where we build many constructs and test them all, and then use machine learning to understand the system from a quantitative perspective so that we can learn and predict the cell behaviors that I've listed on the left side here. So one example in which I've already done this as proof of principle is in the case of CARs. So if you put a CAR into a T cell, when the CAR engages a cancer cell, it'll cause the T cell to kill that cancer cell. And we thought that we could improve CARs by changing the signaling domain, which is shown here in red. And so we used many small pieces of signaling domains to generate combinatorial libraries of CARs with unique signaling properties, and then screen these CAR libraries for their ability to promote cytotoxicity and a long-lived stem-like phenotype in T cells, and then use neural networks to understand how to build better versions of these receptors, and then built a receptor that actually had higher cytotoxicity and higher stemness, and it might be useful in a clinical setting. So to sort of think about what projects I'm interested in the future and what I'd love to talk to people about, I wanted to give two examples of quite different things that I'm thinking about right now. One is in the vein of engineering T cells for cancer therapy. Typically right now, if we use CAR T cells, we put one receptor in both CD4 and CD8 T cells. But we know that CD4 and CD8 T cells are very different. And it's likely that they should have different CARs in the two populations. And so currently I'm doing screens where we're putting different CARs in CD4s and different CARs in CD8s to try to find optimized CAR pairs which would improve tumor clearance. A very different project that I'm thinking about is how can we use engineered receptors and overexpressed transcription factors to reprogram hematopoietic stem cells, the cells that give rise to many types of blood cells. If we do this correctly, then we could guide hematopoietic stem cells into red blood cells at will, which if we pull this off would greatly reduce or eliminate the need for donated blood. I think that would be a huge advance. So find me and talk to me during the session. Thanks for listening.